So now we'll move on with the oral presentations. We have 5 presentations. And the first one is by Verenfridas Knolik. The title is Occurrence of Foodborne Pathogens in Retail Catfish from Bangkok, Thailand.
Good morning, distinguished professor, fellow researcher, and colleagues. Let me introduce my friends and my name first. My name is Verenfridas Knolik. I'm a master student from Chulalongkorn University under supervisor, associate professor Dr. Sarute James Repong. So today I would like to present my research title, Occurrence of Foodborne Pathogens in Retail Catfish from Bangkok, Thailand. So I would like to present this catfish because in Thailand it's like still limited about the contaminated bacteria such as E. coli, Salmonella, and Vibrio in catfish from retail markets. Nowadays, aquaculture have been overcome fisheries because aquaculture have known as the good quality and also delicious based on FAO data. And catfish in Thailand are the second fish that comes after tilapia. Catfish have limited study in Thailand based on the MR study and also antimicrobial resistance about E. coli and Salmonella, which is this bacteria that known as the surveillance bacteria on the aquaculture products. So let's see, these are the productions of hybrid catfish. What is hybrid catfish? Hybrid catfish are the results from the crossbreeding between Clarias gariepinus, which is native in Thailand, and Clarias an African catfish, which is there will get result from hybrid catfish. The mix of this will get the hybrid catfish with delicious of meat and also have a high surveillance, and also the bigger shape compared to Clarias macrocephalus. Another hand also, around 1,252 production in Thailand about hybrid catfish get the GAP or good agriculture practice. And this saw that around 2014 to 2020 that farms in Thailand have grown around 20,000 farms of hybrid catfish. However, there are limited and also the harms of intensive farming to get a lot of productions of catfish, which is in intensive farming way, they could increase the likelihood of disease outbreaks. And sometimes it could lead the farmers to overuse antibiotics. It could lead the AMR, and also this intensive, usually using density catfish in one place of farms, which is that could spread and easier the bacterial contaminations. Another hand, the hybrid catfish for long-term cultivate could let decrease the variety of genetic. It could let the catfish to easier to get illness, which is before the hybrid catfish known as the highest survival. but in the long term, cultivate without update programs, it could lead the catfish to easier get the illnesses and also get infect from some pathogen bacteria. And when the product from catfish reach the retail markets, that could bring some bacteria pathogen such as E. coli, Salmonella, and Vibrio that could harm the consumers who eat that catfish without properly cooking. This my outline research, which is we calculate the samples based on epitools, which is using 50% confidence interval, 95% margin, 95% confidence interval, and 50% prevalence based on E. coli from previous study. And we'll use 10% or margin of error. So the minimum samples should be 97 collected. However, in this research, we will collect 130 catfish. And then after we collected these catfish samples, we will bring to our research laboratory in Chulalongkorn University. And we will take the respective specimens such as gills, mucus, and intestine from each catfish. So we will collect around 390 specimens of catfish. And each specimen that we get 390, it will be detection the three type of bacteria, and following the protocol from BAM and also ISO. And all of the samples positive for E. coli, Salmonella, and Vibrio cholerae, we will calculate using chi-square to measure and determine the significant difference between market locations based on the isolations of bacteria. And all will analyze by SPSS. These are our outline results about each market that we get the contaminants of E. coli, Salmonella, and Vibrio cholerae, which is all of the place of markets have found contaminated by these two pathogens, particularly E. coli and Salmonella. And so the isolation are widespread. It's not localized because all of them not different significantly. And based on the 390 specimens are the total from gills, mucus, and intestine, we found E. coli 243 isolates positive, 243 positive or around 92.3% of E. coli. And Salmonella are 56.2%, and Vibrio are the less, is around 11%. This is our calculation by using chi-square, which is gills show the highest contamination level from every part of catfish, which is compared to mucus and intestines. And for the Vibrio are the less contamination levels, we find that intestine are the less. Based on this, that E. coli and Salmonella are the highest, both in gills and mucus. From previous study, we compare that no detection Salmonella and Vibrio cholerae from catfish specimens from retail market in Indonesia. So in our study, we found from every part of this part of samples. And from previous study also, the gills saw the highest contamination of E. coli, which is E. coli and Salmonella are the particularly important to monitoring for the pathogen contaminations and also MR genes such as blatam and and is the resistant for the third cephalosporin antibiotic. So the percent of E. coli, around 62% are significant in tissue type compared to, and is specific for gills compared to other sample types. And we found that our results are the highest compared to results from Indonesia, which is found E. coli around 21% in catfish samples. And for the Salmonella enterica, we found it's higher around 80% were contaminated from gills and following by mucus. It's also, the result is almost related with another result which is found higher, so it's not significant difference with another study. However, there's research from Egypt, they're found in catfish in retail market, no Vibrio finding. And another result also found that E. coli around 8%. And in our results, we found around 80.3%, and for Vibrio cholerae have found in gills, and following by mucus are around 14.2%. All in total of 3 bacteria are the higher in gills and mucus, because like in the gills are the first occur for against the water contaminates, and mucus as the first defense of the fish. So it's related to that we get the gills and mucus as the higher contamination level. And this is our on progress result, which is we found around 43 isolates have been positive of E. coli, bring out of them 20% are carried multi-drug resistance, and we using by MIC in agar deletion test. And for the E. coli, we found around 4.65% of 43 E. coli carried ESBL with no carbapenems resistance. So our next planning research, we will conduct around, we will get respective isolate that bring multi-drug resistance, will continue to whole genomes by long-read sequencing, and we will do using Oxford nanopore technologies. The conclusion in our study are high prevalence of E. coli and Salmonella enterica are found in gills and mucus, with gills at the highest contamination levels. And it's like raise public concerns, and also the finding of this as the potential biological hazard for consumers. And our recommendations are improve hygiene standard and effective monitoring in local aquaculture practice, and promote awareness on safe fish handling and consumptions, and also encourage broaden antibiotic use following WHO guidelines. Thank you very much. I will appreciate and thanks my lab's mates, including my advisor as a master student. Thank you.
Thank you very much. We have time for a quick question, if we have a question from the floor. If not, can I ask a question? So in addition to actually looking at the health of the fish being sold at the market, have you also followed those fish back to the farm and looked at all the handlers, all the parts in the chain from the, leading to the fish being sold at market?
So actually, our result currently is focusing on the retail markets, but the question actually is make me think for the result, for the future results. I think it could be done. Yeah.
So maybe at the next DAA meeting.
Thank you very much.
Okay, thank you very much.
Just one question. See, when you're telling about the occurrence of these pathogens or something like that in catfish in retail market, have you mapped the entire value chain, or is it specific to the market, or it start from the farm level itself? Whether you have mapped the entire value chain?
So the sample we directly buy from the local markets in like retail markets. So we didn't compare with the farms directly. Yeah.
So you are not sure where the source of contamination is from. Is it only from the market?
Yes, only in retail markets for the contaminations. Yeah.
Okay, thank you.
Thank you.
Okay, thank you very much. Thank you.
Thank you very much.
We're now going to move on to our next speaker, and I invite Gopalan Krishnan Siravaman to the podium, and he's going to be talking about the evaluation of antimicrobial and disinfectant resistance and biofilm-forming ability of Streptococcus agalactiae pathogens from the aquaculture farms in Kerala. So the podium is yours. Thank you.
Yeah, thank you. Respected chairs and distinguished fellows gathered here. I am Dr. G. Keshwaraman, working as a principal scientist at ICRC AFT Kochi, and my work was evaluation of antimicrobial and disinfectant resistance and biofilm forming ability of Streptococcus agalactiae pathogen from the aquaculture farms in Kerala. So all we knows these streptococcosis globally it's affect in farmed aquaculture species, and also nowadays it is as a emerging presence in all the aquaculture farm in Asia, and the streptococcal infections caused by group B streptococcus or Streptococcus agalactiae, and it's posing significant as well as the growing threat to the aquaculture industry in India. The key pathogens include Streptococcus ineae, which is very dominant in Kerala outbreaks, followed by Streptococcus agalactiae and Streptococcus discalaceae and Ictaluri. Generally, these outbreaks often lead to very high mortality, mainly in tilapia fish farming, and these several recurrent outbreaks is taking place in tilapia farming in Kerala. It's definitely because of this outbreak which lead to significant problems, and also it is an increasing prevalence is noticed since the last decade. Then as far as the outbreak is concerned, which lead to almost 45% of the mortality in tilapia fish farming in Kerala, and it is a lot of reports are available. As far as concerned, the Ernakulam district of Kerala, it is reported mass mortality of cultured species, and also they are highly virulent, and it's showing these beta hemolytic activities of Streptococcus ineae. In KUFOS, that Kerala University of Fisheries and Ocean Studies, they did lot of work on the Streptococcus outbreak, and they found that these strains are highly virulent, and also they are multi-drug resistance. Generally, the Streptococcus infections in aquaculture farming, mainly tilapia farming, which lead to lot of mortalities and severe economic losses to the farmers, mainly by reduction of this total production and increase the treatment cost, as well as the marketability of affected fish is reduced. And all as we know, quite naturally, several risk factors are attributed for this outbreak of Streptococcus infections, mainly the temperature, pH, and salinity. It's giving wide range of this temperature, pH, salinity, as well as nowadays the high stocking intensities and elevated water qualities, as well as the ammonia levels, mainly contributing for this outbreak of Streptococcus infections. And all we know, this Streptococcus, group B Streptococcus agalactiae, is mainly of one health significance. It's affecting fish as well as the human. All we know, it's affect the fish, and it causes meningoencephalitis by affecting these nervous condition. That's why we can found that abnormal swimming, and also it's cause skin infections, which cause septicemia, and also it affects an eye and causes ocular lesions such as exophthalmia and corneal opacity, and also skin lesions. And in case of the humans, so it affects these maternal sites and cause meningitis, septicemia, encephalitis. And it's affected during this initial stage of birth, and its streptococcus infections mainly occur through this entry up through the reproductive system of the females, and also cause similar kind of infections in babies after birth. Also it will cause septicemia, meningitis and all. So keeping in view of this in One Health perspective, so we carried out these objectives: isolation and identification of Streptococcus agalactiae from farmed fish in Kerala, then evaluate the level of antimicrobial resistance, then assess the pathogenicity of Streptococcus agalactiae infections by biofilm forming ability, then evaluate this disinfectant resistance and also screening its resistant genes. So these are the works we carried out in Kerala. We covered the 2 districts, Thrissur and Alappuzha, and where mainly these aquaculture farming activities is taking place. We collected 100 samples each. That is total number of 206 samples were collected. And this is the protocol we follow. We isolated and we identified. We collected including the water sample. We want to observe what the potential source of these infections are spreading through the different forms. Then we identified by using protein culture-based methods as well as the 16S rDNA, and also we used automated identification system to confirm these Streptococcus agalactiae infections. And then we evaluated its antimicrobial resistant pattern, then disinfectant resistant and biofilm forming activities. And we established the protocols for this isolation of Streptococcus agalactiae infections, and we collected the samples. Then we used an 5% SRPs and TSA plate, and it's showing typical beta hemolytic on this TSA agar containing 5% SRP, and such colonies where we collected. And then we confirmed its group B Streptococcus by Lansfield Histostrip latex kit, and then we used 16S rDNA also, and then we used it PD Phoenix automated system to confirm it. Once it is confirmed, then we phenotypically evaluated for this antimicrobial resistance, disinfectant resistance and biofilm forming activities. And then based on these phenotypic characters, then we evaluated for these genotypic levels to antimicrobial resistance, pathogenicity as well as this biofilm forming ability. So these are the ways by which we identify these ARGs, antimicrobial resistant genes, disinfectant resistant genes, and also the biofilm forming activities. These are our results. So we identified as a gram-positive bacilli, non-motile, beta hemolytics, and 5% C4BC TSA agar, and it can able to grow at 30 degrees centigrade, and also that 6.5% NaCl, and utilize pyruvate as well as the cuprate. That is the specific test for the confirmation of Streptococcus agalactiae from the other groups, and also it utilized the glucose to produce the acids. Then we used Streptococcus species-specific primers covering the 16S to 23S RNA intergenic space regions. So our results, we found that total 6 number of isolate was confirmed as a Streptococcus agalactiae. There is the incidence is around 3%. Whereas the Alappuzha district, it's having the higher numbers as compared to this Thrissur district, because the backwater was very, very closely covered in all aquaculture farms as compared to Thrissur districts. In general, we used to carry out this active AMR surveillance. Our CAFT Institute, we are the nodal for project also, and we keep on used to do active surveillance in all the aquaculture farms, and we established the protocols for the isolation of AMR pathogens. And this protocol, we used it by these automated identification systems for these gram-negative bacteria as well as the gram-positive bacteria. and selective enrichments in the presence of antibiotic, and then we will selectively isolate and straight away we will go for the automated identification system, and this system can able to give the alert if it is beta-lactam is producing Enterobacteriaceae or methicillin resistant Staphylococcus aureus, vancomycin resistant Streptococcus. So based on these results we taken in these studies only on Streptococcus agalactiae, because based on this antibiotic resistant pattern, and then we screened for the about 6 number of Streptococcus agalactiae we obtained, and also the uniqueness is we found 6 more isolate of Streptococcus porcinus. We all knows these are the human pathogens, and its presence is noticed in the aquaculture farming also, so it is clearly indicating that there is a contamination from the hospital settings also possible in the aquaculture farming. So in these studies we took only these 6 number of Streptococcus agalactiae, and we went for further antimicrobial resistant pattern, though we employed around 6 number of antibiotics, and we could found that distinct antibiotic resistant to cefotaxime, chlorophenicol, tetracycline and levofloxacin. But this is the unique kind of resistance we observe it. This is about this diffusion methods showing that it is a level of resistant to these potent antibiotics, and of course we are aware that this cefotaxime resistant is not common in group B Streptococcus agalactiae, and this could be due to this contributing factor like horizontal gene transformation may be possible, as well as the cefotaxime resistance. The another important resistance is due to these clindamycins. This also very rarely reported, and it is considered as a growing concern, and this resistance mainly contributed by this presence of ERM as well as MEF gene family, so which definitely lead to the development of multi-drug lincosamide streptogramin B phenotypes. That is, this could be a inducible clindamycin resistance. So this is a serious concern, but it is not very often reported except in China. But this presence recently we noticed in India as well as. So similarly we could found for this levofloxacin resistance. And this we are aware that levofloxacin resistance is mainly due to these point mutations in this gyrase A gene as well as PARS G genes. Because of this minor alterations in one amino acids, which lead to this entire change of these regions to cause for the resistance to levofloxacins. And this is also one of the emerging issues, so we should take care. And this resistant mechanism may be due to this unknown resistant mechanism or due to the any novel resistant pattern. And also it's commonly showing resistant to tetracycline as well. Then we evaluated phenotypically for this different level of most commonly used disinfectants, like 5 numbers, and we found the resistant to higher level of MIC2, chlorhexidine, benzalkonium chloride as well as CTAP. Then we, by multiplex PCR based, we evaluated for the resistant genes, and we almost 6 isolates they show this resistant to many of these disinfectant resistant genes, and 3 isolates shown 5 resistant genes, and 2 isolates found positive for 4 number of disinfectant resistant genes. And we evaluated for these biofilm activities. We all know this is directly contributed for these it is go resistance with this antibiotic resistant pattern, and we could found that 3 of the isolate were potential biofilm producers. And this is about the nutshell of this overall result of these present studies. So we can see this antibiotic resistant pattern. It is high in many of the antibiotics, and also with the recent antibiotics, it shows this disinfectant resistance. They are potential biofilm producers and also acquire these strong virulence resistant genes. So by this I would like to conclude that despite the presence of low incidence of Streptococcus group B infections in aquaculture farming, but so the higher level of resistance, it limits the effective treatment. And significantly the AMR pattern was higher in compared to these routinely recommended antimicrobial agents in case of aquacultures. And also it is strong beta-hemolytic activities on sea blood agars, and it's showing potential biocide resistance, and also biofilm forming capacity of this one enhance the persistence. So the Streptococcus agalexia poses a hidden threat because it's having multi-facet level threat to aquaculture industry, and it will pose significant threat in terms of One Health. And also with this, definitely the research came, I want to say that viewpoints, anyhow, there is no standard SOPs in terms of we able to identify these, all the Streptococcus species, and lack of protein diagnostics also available, and limited awareness in terms of One Health to the farmers. So keeping in view of that, so we must need the integrated AMR surveillance in One Health perspective, and also revisiting farm level disinfection protocols, and also we have to development of biofilm targeted intervention in One Health aligned strategy. Thank you.
Thank you very much. A huge body of work there. Do we have...
So it's very interesting and also disturbing that you have you have resistance to even the disinfectants. So do you have any suggestions of how one could deal with that? Because we have lots of work going on AMR, but to deal with these genes, and what is your idea of a strategy that would be to cope with this?
Yes, madam, good question. Madam, actually we go to, nowadays we all knows in case of the antibiotic treatment also, no single drug is effective. So we go to try similarly for these disinfectant resistance. So we try to see it, either we must enhance these activities of existing disinfectant by including phytochemicals. That could be good strategy. Otherwise, we can see it continuous evaluation of resistant to disinfection also possible.
Or a mixture of disinfectants, as you are saying, not just one, but more than one, to mix them and use them? Different kind of disinfectants?
Yes, ma'am, that's what we have to see, because we have to target different facets of these resistant mechanisms.
Thank you.
Thank you.
Thank you. One more question.
Hello, Dr. Shivran.
Yes, sir.
Nice talk.
Thank you.
I wanted to know whether there are any international standards available for telling us that this disinfectant is resistant, this microbe is resistant to this disinfectant. So, for example, in AMR, there are CLSI guidelines or UCAST guidelines.
Yes.
So, for disinfectants, whether there are any internationally accepted guidelines to claim that this is resistant or susceptible?
Yes, sir. They have given in MIC.
MIC, who has given? See, you have told us that this triclosan is resistant or susceptible. But what is your criteria to declare that it is resistant or sensitive?
Sir, routinely we can see it, the existing disinfectant concentrations, commercially available concentrations. Okay, 0.1, 0.5, maximum 1 microgram per ml is permitted, normally available. So when we see it, these ones are above that level, so we can say that, so it's showing this higher MIC.
Thank you.
Thank you.
Thank you.
Thank you, Dr. Sivaraman. The next presenter is Mr. Andrew Lee Powell.
Thank you.
The topic is setting epidemiological cutoff values, Vibrio parahemolyticus, a One Health case study. Mr. Andrew, please.
Hello, everyone. I'm Andy Powell. Nice to meet you. I'm here representing the UK FAO Reference Centre for AMR. This is a tripartite with 3 government organisations in the UK, the Centre of Fisheries, Aquaculture and Science, Animal, Plant and Health Agency, and the Veterinary Medicine Directorate. And together we hope we provide a One Health service to tackle AMR globally. So today, I also want to mention, that we've got a stall at the back of the room
So please come and say hello to us if you have time today. Sorry, is it the green button? So the situation of vibriosis in Europe is of growing concern, particularly with climate change. So in Northern Europe, we call this the crucible of climate change because the Baltic Sea temperature is rising at an alarming rate, and we're seeing increased outbreaks in humans over the last 25 years or so. So it's a big concern, and the ECDC rate this as one of the highest risks according to climate change. Ooh. So we know, as everyone, many people have this, that it's a big problem in aquaculture, and not just the shrimp industry, but also the finfish industry as well. So, the O'Neill report. So, this was a big report that came out in 2015, and it recognized that AMR is a massive problem, and kind of quite a big shock to everyone that AMR is going to cause many, many deaths, 10 million deaths by 2050. So this is a big reference point for us in the reference center. The CDC, that have very good epidemiology around vibriosis, predicts that they have 80,000 cases per year. In Europe, non-O1 cholera vibriosis, non-reportable. So the epidemiology for us is a little bit sketchy, and it's something that we really need to work on. Vibrio parahemolyticus is a human and animal pathogen. In humans, it can cause gastroenteritis, wound infections, and sepsis, mostly through the consumption of raw seafood, but also interaction with the sea as well. So we know that there are many kind of superficial ear infections and wound infections that go undiagnosed and unreported. It's a massive problem in aquaculture, and the big problem is there are currently no expert rules for many of the aquatic pathogens, including many of the vibrios. So AMR surveillance in aquatic animals. From this table, you can see this is taken from the CLSI guidelines, and there are only 4 entries for aquatic pathogens. So part of our role of the FAO AMR Reference Center is to readdress the balance and get some of these aquatic pathogens into the expert rules. So between the 2 main conventions for AMR, the CLSI guidelines and UCAS, they use different acronyms for setting epidemiological cutoff values. They use the acronym ECOF and ECV. Effectively, they are the same thing. They are referring to non-acquired resistances or wild-type bacteria. And this means there's no phenotypic response. Also, recently published are the regional guidelines that were published between FAO and Singapore parks. This is a very useful document, and I recommend that you all take a look at this. So, why do we need these rules, and why do we need to work together to gather the data to set these rules? So you need at least 5 laboratories providing data. For our study, we had 8. A minimum of 100 wild-type isolates. So if you find you don't have expert rules, you can or you can set your own local epidemiological cutoff values by looking at 20 wild-type isolates. Each lab must test 20 isolates, and the lead laboratory needs to provide a protocol to make sure that everyone is following the same methodology and using the right control material. And you need to agree the diagnostic method. So I think particularly with Vibrio, it can be a bit of a challenge. I think we lean heavily on the modern techniques such as 16S and Moldy-Toff. In many cases, they don't give accurate results. We know through some of the testing that we've done on environmental samples where we've done kind of reverse diagnostics, if you like. So you sequence first and then try and see if the traditional methods and the secondary diagnostic methods match up with what you found through next generation sequencing. And quite often they don't match, and that includes the published PCR sequences. So you need to be very careful when you're trying to identify some of the pathogens in the Vibrio genus. Okay, so just to be clear, we produced 2 papers here, one on disk diffusion and one on MICs at 2 different temperatures. We supplied many of the materials to the participating labs. This was a logistical challenge. And we provided a data sheet for reporting. This is really important so we could weed out any of the data where the control results didn't match what was reported in CLSI. And also you may need some contractual agreements. This is kind of very boring, but when you come to publish your material, if you don't have these agreements with the institutions you're working with, it may hold up your publication, sometimes by many years. So we used ECOF Finder for this, and it's an algorithm that is freely available on the CLSI website. It's no longer available on the UCAS website. However, for the purpose of this study, we use this algorithm, and it will tell you whether your isolates are wild type or non-wild type, so you can take out any of those non-wild type isolates when you're setting the epidemiological cutoff values. We also used NRI, the normalized resistant interpretation. Again, this is a freely available algorithm that you can use, and this compares data across the data set. You can compare it between labs, and it enables you to do the quality control. Now, when we had the data together, we had to present it to CLSI, to the Aquatic, the Antimicrobial Working Group, AWG. And this is an example of how we presented it to them. We have a slide for all of the epidemiological cutoff values, and we also have histogram as well. They like to see it presented like this, where you show where the cutoff value should be. And this is for fluorophenical at 35 degrees. And these were the proposed cutoff values that we presented to them for the MICs. I think it's just worth quickly mentioning some of the issues that we have with disk diffusion. 3 of the 5 participating labs had some issues. One of the labs had 93% even zone sizes, and this we think was because they were using like a ruler rather than calipers. We use digital scales. Sometimes you can get some sliding with the ruler. So we've not only set epidemiological cutoff values for Vibrio parahemolyticus, there are other pathogens, and during this process we worked with 14 countries and 29 institutions, showing the emphasis on collaboration. We've also worked with American labs and labs in Asia for this. So in summary, the objective was to find some ECVs for Vibrio parahemolyticus at 2 temperatures. We had 8 labs involved. For the MICs, many of the results were consistent for both temperatures. And when we compared the values statistically, 56% of the 1,473 results were identical, and 38% of those were differed by not more than 1 dilution step. So in the disk diffusion, as expected, there was quite a big difference between the 2 temperatures that we tested. And we had 1,314 paired observations. And these are the cutoff values. I'd quickly like to thank the organizers of this event and the staff as well, who've done a really good job here. We worked with many different institutions during this process, across Europe, America, and Asia. And a big thank you to Pete Smith at the University of Galway, who's been a real champion for AMR in aquaculture over the years. The publications for MIC you can get through looking at this QR code. The data, as I said, has been presented to CLSI and will be in the next version for all field vets to use. So thank you very much.
Thank you, Andy, for a very clear presentation. Do we have a question from the floor, please? Yes, Celia.
Thank you very much for the very clear presentation. Do you think salinity would be a factor in doing MIC with the marine isolates?
So we follow the guidelines that are set in CLSI, and it does state that you can use Muller-Hinton agar for this. Whether salinity plays a part or not, that's something that we would have to test. We know in the environment it plays a big role in the outbreak or the prevalence of Vibrios in the environment. So, yeah, it's a valid question.
So, would it be okay to add salt to Muller-Hinton when you do it?
I think you need to follow the guidelines. So, it doesn't, for the Vibrios, as far as I can remember, I don't have a photographic memory for the guidelines because they're quite extensive and nebulous in some cases. So I think you need to refer to the guidelines and you need to follow the guidelines. And if it doesn't state that you need to add salt, then you don't have to add salt.
Thank you.
Thank you.
And another question from...
Thank you very much for the wonderful presentation. I just have a curious about, do you have the plan to link between genomic data with the wet lab data? So you'll be able to determine or develop the program that can predict AMRs based on genome? Do you get what I mean?
Yeah, I think I'll speak to you afterwards about this because I couldn't hear the question properly, but I'll come and speak to you in a minute. It's tea break. Thank you.
Okay, Andy, thank you very much. And again, if we could just thank Andy, thank you. Oh, I'm sorry, we have one more.
Too late, I'm gone.
Sorry, we have one more, Andy.
Yeah, no problem.
Hi, thank you. Great presentation. You were mentioning about the multi-TOF and 16S sequencing for the identification of the agents. It just stresses the impact of building the database with more information and sharing this information, because at our institute, my colleague has been building the multi-TOF and we are sharing it freely with all the private labs in Norway as well. But I guess what we really need to do is actually share it across the countries. But I don't know how we can do it. I hope you can.
All of the papers that we produce through the reference center are open access, so they're free for everyone. In terms of some of the methodology that we're developing at CFAS, so one of the assays that we've developed is a nanopore sequencing, where we are sequencing a single gene, very much like 16S, and we've created a huge database, and it seems to be very useful for speciating Vibrios particularly. But we will publish this and we will make it freely available to everyone. It's not an off-the-peg assay, but it's very useful. So watch this space.
Thank you.
Okay, thank you very much. Thank you, Andy. We're now going to move to our fourth talk in this session. And if I could invite Priyabrat Swain to the podium. And they're going to be talking about the risk of escape pathogens in aquaculture of Eastern India. So the floor is yours. Thank you.
Good morning, respected distinguished all delegates. The topic is risk of escape pathogens. We know escape pathogens belongs to 6 genera of bacterial pathogens, which are very, very prominent and multi-drug resistance, and they are causing a lot of risks for human mortality as well as death across the globe. So we have studied what is the incidence rate of escape pathogen in aquaculture. We are working with 7 medical institutes and veterinary colleges. Dr. Karuna Sagar already, professor, told that what is antimicrobial resistance and what is the importance of significance of aquaculture in AMR also. And mostly misuse and overuse of antimicrobials contribute to development and spread of antimicrobial resistance. And antibiotic resistance bacteria has emerged due to entry of some of the antibiotic in aquatic environment through sewage and urban discharges. And AMR in aquaculture presents a serious ecological and public health concern, so as medical institutes are concerned. Most commonly used antibiotics in aquaculture worldwide are 8 to 9 antibiotics are mostly used, even in India, some of them are used. Approximately 80% of antimicrobials used in aquaculture enters to the environment. And escape pathogens are mostly, they belong to 6 genera of the bacteria, and they are multi-drug resistance. And here, besides escape pathogen, I have taken one Aeromonas hydroplasy fish bacteria pathogen, since it is having a zoonotic importance, and a reference bacteria also. Our main study to determine the prevalence of AMR bacterial isolates from aquaculture, particularly escape pathogens, including Aeromonas, and to analyze the resistance pattern and phenotype among these different bacterial species. The study mostly, it includes a 7-year study, 2018 to 2025. And we have covered mostly 2 states of India, which are major aquaculture states of India. And in each state, we have covered 7 to 8 districts. And these are the samples given right side. And we also do use chemotherapy antibiotics used in aquaculture, but very less. These are the drugs, chemical formulations used in farmers in Indian aquaculture. And most of the common antibiotics used are OTC, oxytetracycline, doxycycline, enofloxacin, cephalexin, and erythromycin. These are already used. And from April 2018 to March 2025, we have collected several samples. Total bacterial isolates is 3,961, very huge data. It includes E. coli, Staph aureus, and Aeromonas species. And we have taken samples from fish as well as pond water. And if you can see the resistance pattern, particularly Aeromonas in fish of Odisha, particularly one state, you can see most of the resistance in cefotaxime, which is a cephalosporin third generation antibiotics, and it is highly resistant. And resistance pattern Aeromonas from pond water of Odisha, it is also same pattern like fish. And resistance pattern of E. coli from fish in Odisha farms, you can see these are the patterns. And the resistance also, nalidic acid also has got resistance, some resistance, and ampicillin. Resistance pattern of E. coli from pond water of Odisha. So these you see again the cefotaxime is a highly resistant. Staphylococcus from fish farms of fish in Odisha fish farm. These are the resistance pattern. Penicillin group is more resistant, and cefotaxime, and also erythromycin. Resistance pattern of staphylococcus from pond water, and you can see the same pattern, cefotaxime and penicillin and erythromycin. So there is absolutely no difference between pond water and fish. And if you go to another state like Andhra Pradesh, so this is the resistance pattern. Motilely there is these are the patterns, cefotaxime, same, mostly cephalosporin group antibiotics. Resistance pattern Aeromonas from Andhra Pradesh. So this is also the resistance pattern, mostly resistance to ampicillin and cefotaxime. Resistance pattern of Escherichia E. coli from fish farms of Andhra Pradesh, and you can see the resistance in ampicillin and cefotaxime. So these are the resistance pattern you can see very well. Even the resistance occurs in tetracycline and enofloxacins. Resistance pattern of E. coli from pond water in AP farms, Andhra Pradesh farms, and these are the again ampicillin, and these are the varying and mostly pattern. Resistance pattern of staphylococcus from fish farms, Andhra Pradesh, you can see again penicillin, and here it is cefotaxime. So cefotaxime, penicillin both are mostly causing resistance. And resistance pattern of staphylococcus from pond water of AP fish farms. So again, this is cefotaxime and penicillin, and here erythromycin. So if you see the resistance pattern of Aeromonas from Odisha and Andhra Pradesh, so this is the most resistance is cefotaxime and amoxicillin group, and these are the both states. Resistance pattern of E. coli from Odisha and Andhra Pradesh, particularly fish farms, you can see these are the fish farms, particularly from Odisha, that is from both Odisha fish water and fish samples. You can see the resistance pattern is ampicillin, then cefotaxime, and like that, and tetracycline, also nalidic acid, enofloxacin. And one of the escape pathogen, Klebsiella, is a very important pathogen, and you can see the resistance pattern is not much more particularly in aquaculture concern. Resistance pattern of Acinetobacter is another human pathogen, and you can see the resistance pattern. So resistance pattern is more in Acinetobacter. This is a bacteria mostly found in the human skin, but it has emerged as a very important pathogen of nosocomial infection. Maximum human death in ICU and ventilator patients occur with this, and some resistance has been developed, particularly Acinetobacter and most of the antibiotics. Resistance pattern of Enterococcus species, we can also see it is not also alarming, except Acinetobacter, one bacteria, others are not so much alarming. Resistance pattern of Pseudomonas, some is there, but it is not in an alarming state. And if you see the use versus resistance of different antibiotics against escape pathogen in Indian aquaculture, all the data has been analyzed by AIMS Bhubaneswar in Vitech system. You see the used, that is 5 antibiotics are commonly used, and those are not used. But if you see the comparison, even if they are used, the resistance is not more. But those antibiotics are not used in aquaculture, resistance is more. So it seems that aquaculture is a victim, not though it never contribute to the development of AMR. Rather, the antibiotics, residue antibiotics from other sources goes to the aquaculture, and the bacteria has been resistant. So this is not fault with our fisheries and aquaculture people or aquaculture business. And in the summary, you can see the antibiotic total 3,000 961 bacterial isolates sampled from 851 farms of Odisha and Andhra Pradesh. And E. coli isolate found resistance to ampicillin, followed by tetracycline, nalidic acid, and ciprofloxacin. Aeromonas highest resistance is ciprofloxacin, followed by cefotaxime and trimethoprim and amoxicillin. For Staphylococcus species, highest resistance was found in penicillin, followed by erythromycin, cefotaxime, and tetracycline. To the escape pathogens, Enterococcus species is resistant to erythromycin and tetracycline. Klebsiella was resistant to imipenem, meropenem, and ampicillin. Acinetobacter is found to 100% resistant to many of the antibiotics. So Acinetobacter is a problem in water now. So it is mostly resistant to all of the antibiotics. And Pseudomonas species are resistant to microcycline and tazobactam and all. These are the antibiotics. So it seems, except the one bacteria, the Acinetobacter, other bacteria are not so much alarming situation. And aquaculture does not pose any serious threat, particularly to the AMR is concerned. But we should continue the survey, and medical institutes want the water and fish should be all the time surveyed to help the WHO, FO as well as INFAR of our country. So in the conclusion, the study indicates, I can say, aquaculture situation is not so alarming in AMR, but it is a victim to the AMR, I can say, because most of when the things comes, the people say that India or any other countries, there is a lot of ban, lot of people don't prefer the antibiotics containing fish food, but India is still, I can say, it is a safe. So collaborative efforts across the government, researchers and communities are essential to combat antibiotic resistance in Eastern India's aquaculture sector. Mitigation strategies include surveillance, monitor antibiotic use and resistance. It should continue. Policy and regulation enforce structured guidelines of antibiotic use, which is under process by Government of India and other countries. Educate the farmers to use alternative to probiotics and vaccines, because some of the vaccines have been in the pipeline, some are in the, now people are concentrating more on probiotics and vaccines. Invest on sustainable fish health management and public awareness. It is very, very important. And I must acknowledge the study was done by ICR, Department of Biotechnology, because the study was been done last 7 years, including 3 to 4 projects from different, like Department of Biotechnology, Indian Council of Medical Research, Incline Trust International, and of course our host institute, SIFA. Thank you. Thank you all.
Thank you for a very clear presentation. Lots of very interesting information in there. I invite questions now from the floor if anybody has a question. If not, can I ask, what's next? How do you take this information back to the farms? How do you now effect change?
Next challenge. So our next challenge will be we have to go on surveying the antibiotics. First thing, the AMR pattern. Second thing, regulation of the antibiotics, which is in force now. We are having one team, expert team, working in another project. We are just streamlining what is the use dose and use of antibiotics in our aquaculture. Then second thing, we have more awareness to contain this AMR spread. And third thing and fourth thing, we should tell our audience, to our people, that still fish is safe to consume. It is not a problem now. Thank you.
Thank you. Thank you. Any other questions? If not, then thank you very much, and we'll move to our last presentation. Thank you.
So the final presentation will be by Wafas Beit Al-Arimi. Overview of the National Plan for the Development of Aquatic Animal Health Project. Ma'am, please.
Thank you. In the name of Allah, most gracious, most merciful. My name is Wafa Al-Aremi from Aquaculture Center in Oman. And first of all, I'm honored to be here to share my information and giving you an overview about overview on the national plan for the development of aquatic animal health. So the Sultanate of Oman has identified aquaculture as one of the key pillars to diversify its national economy. Therefore, Ministry of Agriculture, Fisheries and Water Resources is engaged with several research and development projects, such as choosing suitable sites, selection for aquaculture, hatching and culturing of different species, such as shrimp, finfish, mussels, oyster, abalone, sea cucumber, and others. Development of freshwater integrated tilapia farms, development of feed of cultured aquatic animals, and others. And this work is done not only by, it is done by different departments and entities within this ministry, such as Aquaculture Center, the Department of Aquaculture Development, Food Safety and Quality Center, and there is a new entry of the General Directorate of Animal Health, represented by a new established department of aquatic animal diseases, and I believe this department will take the lead in dealing with farms and this industry. Also, we have FDO, Fisheries Development Oman, that contributed a lot through its having different companies that also add to this industry. So as a result, you can see there is an increase of production of aquaculture and aquatic animal, and mainly because of production from shrimp of 62%, followed by sea bream 29%, and then by tilapia 9% over the last year from 2019 to 2020-24, of a production of a total of more than 5,000 tons. For example, if we speak about farmed shrimp, you can see that such as the venami, Penaeus venami, there was an increase of the production from 2022 to 2024. And also we have, even though that we have several shrimp company, but there are only main two that they produce, they give a production. And regarding sea bream, there are three commercial companies that use the floating cages on the sea, and there are increased production as well. And the last one is the Nile tilapia. And you can see, even though there are more than, let's say, 26 companies, but you can see that there is a humble production, and that because they are facing some kind of challenges. And here the role of Aquaculture Center and any department involved in aquatic animal health, they should play a role in developing these farms. And also for us to keep up this growth and to make the figures going on and on, there was a necessity to propose a national plan for the development of aquatic animal health that consisted of 6 programs. For example, program number 1 is the development of a specialized laboratory in aquatic animal health. And I joined the Aquaculture Centers in March 2023. And the first thing we did is to develop a laboratory to enable us to do our job as best as we can. So we started from a room, as you can see in the lift. And then we did a lot of work regarding tender analysis, and finally we got this laboratory that inshallah by the end of this year we will be completed and we will open it. And the purpose of this laboratory is to do research, and we will be able to do different things, including sample preparation, molecular, bacteriology, parasitology. Regarding histology, which one of the sections that I will not sacrifice, we have another adjacent building. They already have histology, and we got their approval for us to use it for our detection purposes, but it needs a little bit adjustment to fit our purpose. Regarding program number 2 is contribution in biosecurity and management of aquaculture farms. So as you can see, what is our role is to participate with other bodies like the Department of Aquaculture Development in visiting these farms to do inspections. And in this inspection, we are trying our best to give them whatever knowledge that we have for a better management and in that farm. For example, we can verify whether they are implementing biosecurity measures, whether they are following the regulation, whether also if they have any problem that we can solve at that moment, or we can recommend ways to solve these issues. Also, we give them some advices regarding good quality of water, because it is very important, and also the importance of recording. We sometimes give them a ready-made draft of these forms that for them to fill, and it will also help us in the future if there is any case of mortality. We will be able to detect what is the source of problem. And also, sometimes we give them training on how to handle fish, or how to do cleaning for the tanks. Many of the problems of death or mortalities was because of, let's say, poor management. And also, I remember Dr. Shin in his slide, he mentioned because of increase of the temperature, there is like more chance of mortalities because of bacterial infections. That's why we give them some hint to do some kind of roofing to decrease the temperature or to lower the temperature, and others. So regarding the program 3, detection of aquatic animal pathogens in farm and wild animals, of course we need to be concerned about multiple of fatal pathogens in fish, crustacean, and mollusks, especially those are listed in OA. But so far there is no reports that we have this kind of pathogens in our water, but that doesn't mean that we don't need to do some kind of surveillance, monitoring, and field-based surveys to make sure they are not really in our waters. And I believe this job will be taken by the new emerging department under the Directorate of Animal Health, and we will help them as long as they will need our help in this. Regarding program 4, which is contribution in the early warning programs and responding to the mortality cases, as you can see, while we are waiting to complete our work in our laboratory and make it ready for us to go on, we were doing some kind of responding to some of the cases that we get from wild and also farm. For example, there were few, but we try our best to help. So there was some bacterial infections in farm, and there's some also parasitic infection in some of the aquaculture facilities like aquarium and so on. And we did the analysis for diagnosis only based on what we have, in the time being. So we did like from the clinical symptoms, also from the bacterial isolation or from microscopic examination. And we kept some samples for later to do continue analysis using molecular. Regarding the 5th program is regarding enhancement of the economy and investment by supporting innovation and technology in the field of aquatic disease. I believe, as being the R&D, Research and Development Center, our job to be to provide help in prevention, detection, and treatment. So inshallah, in the future, our focus will be in optimization of method for diagnosis to help other institutes in their job, especially now Oman is open to, would like to send their product to other markets, the international markets, and one of their requirements is to screen for those OWA listed diseases. So since that we have experts in using molecular methods, so we are willing to help them in optimization of these methods. Also, we submit proposals regarding development of rapid laboratory testing for screening. We submit a proposal for the use of alternative treatments to reduce the antibiotic use in farms. Also, and there is, we would like to participate in future vaccine programs in the area. Regarding the sixth program is the training of Omani's staff. So we are not only concerned to train our staff in the molecular and different diagnostic methodology. We also would like to transfer our knowledge to the other department in animal health and other department in food safety and so on. So we started to do this kind of a practical training. For example, in 2023, we did like the regional workshop of aquatic animal health of the Ayora region. And in this workshop, we focus on biosecurity measures in the farm. And we did a trip to one of that farm that we felt that a little bit better. It is like we did some rule in that development of this farm. In 2024, we did like a practical training, detection of viral infection in shrimp. And in 2025, we did the training, the similar training, but not this time not in our building, but we reach the other people. We go there and we use their facilities and laboratories for them to make sure that everything is working well. So in conclusion, we are hoping to continue doing our job in strengthening the capacity and assist the relevant authorities in implementation of biosecurity and health management in aquaculture. Also, as I said, as R&D center, we were working developing diagnostic methods to help relevant laboratories. And actually, we started with one of these pathogens, and hopefully we will continue for the other. Continue knowledge transfer to the other related bodies through practical training in disease diagnosis. And also, we aim to improve, we will focus in improving the prevention methods and enhancing the use of alternative treatments for pathogen in farm. At the end, I would like to thank the Ministry of Fisheries, Agriculture, Fisheries and Water Resources and the National Program for enhancing economic diversification, Tanfeed, for funding of our project. It is like a carry-on project, so it is renewed. Every time, every 5 years, it renews, and we hopefully it will continue so. Also, I would like to thank our director, Engineer Khalfan Al-Rashdi, for his support, and the team of the Department of Aquatic Animal Health. And finally, I would like to thank the organizer, especially the convener of the DAA 12, for giving me this opportunity to share our short story about what's going on in our project. Hopefully, in DAA 13, there will be more findings to present, inshallah. Thank you.
Thank you very much, Dr. Wafa, for a very, very nice presentation. Thank
Thank you.
Do we have any comments or questions from the floor? Oh, I see a sea of hands going up. Ed, I don't know if you want to go first and then...
Thanks very much. No, thanks very much. Really good presentation and great to see the growth of aquaculture in Oman. You mentioned that Oman is free of OIE, WOA listed diseases. I wondered if you could tell us a bit about how the government works with industry to maintain that disease-free status.
Again, could you repeat your...
So what's being done to ensure that you keep free of these WOA listed diseases?
Yeah, the thing that I said that Oman is, there are no reports of this, pathogens in our water because we didn't do any, till now we didn't do any kind of surveillance or surveys, but we actually, of course in the future we should do to make sure that Oman is a clear zone. So, and our data that we have so far is based only in emergency calls, but inshallah we will do this kind of surveillance to make sure that there is nothing. For example, we have, as I mentioned, that we are collaborating with the Department of the Development, and this department is going to do an inspection in farms, and according to the farm's records, there is no evidence of having this kind of pathogen in the farm, and we didn't get any reports from the wild that there is any kind of this issue. So only my answer depends on the data that we have, but of course there will be surveillance and surveys in the future to make sure of this finding. Thank you.
Congratulations on setting up a wonderful lab. Just wanted to know whether you are also looking at international collaborations in training or some setting up, say, quarantine centers, like things he was saying. How will you ensure that these diseases do not come to Oman? So whether you will have these quarantine centers or some checks and all. You were mentioning surveillance, of course. So whether you're looking at any international collaborations too?
The thing is that, you know, as I said, there is an entry of a new department. It's from Directorate of Animal Health, and I believe they will work hard or maybe harder to make that kind of planning, you know, how to deal with this, because, you know, as I said, Oman is want to go to the markets, and they need to do this kind of surveys. So they will, I believe, when they will do this planning, they will gather all the people involved in this aquatic animal health and aquatic health disease. We will be coming together in one table and discuss how we can prevent entry of this kind of pathogens by strengthening the diagnostic tool in the quarantine facilities. Also, we will encourage the surveillance, we will encourage the monitoring of the farms, so to make sure that nothing go in and nothing also the diagnosis before the export. So nothing go in, nothing happen in the middle, and also nothing go out. Yeah, thank you. There will be collaboration, inshallah. Actually, we have a focal point, OWA, from OWA. We have a focal point in Oman, in which that we sometimes, if we find something, we report to them. And actually, I would like to use this opportunity, because one of the challenges that faced us and delay our progress in doing optimization is that we don't have the positive controls, you know, for doing this optimization. So if anybody, I forgot to put my email. So if anybody have this kind of positive materials for any of these OWA diseases, pathogens, please help us. We want to progress more. Thank you.
Thank you very much, Dr. Wafa. Thank you very much. Thank you. Well, ladies and gentlemen, that brings us to the end of the current technical session. May I thank all of our speakers once again. Can I also thank all of the audience for your enthusiasm for the questions. And I think tea awaits us. So see you for the next session. Thank you.